2 minutes, sir.
Yep. His name is Suresh, right? N. Suresh. If our body is a country, then microbiome is a very important state. Hello and welcome to BioTecNika, and today we are celebrating World Microbiome Day, powered by Association of Biotech Led Enterprises. Namaskar, my name is Shekhar Suman. Welcome to the session, guys. Welcome, everybody. Today I am privileged to have a great lineup of speakers who are going to not just enlighten us about microbiome, but we are going to celebrate microbiome. My dear followers, fans, and critics of BioTecNika, in all our endeavors to promote biosciences, we must celebrate World Microbiome Day. Well, you ask why? The answer lies in the question itself. We won't exist in a healthy state if microbiome around us doesn't exist. Well. We all have been traumatized by the COVID-19 pandemic and now whatnot, monkeypox or whatever viruses we are seeing around. But you look at this. If there are good, then there is evil. And microbiome is the good around us. Let us not just celebrate or try to promote microbiome. Let's be proud of microbiome. Let's enlighten everybody around us about microbiome, because a lot of diseases can be cured. A lot of work can be done with the help of microbiomes. So today, to enlighten us, we have a vast range of speakers with us who are going to give us some insightful thoughts on this particular topic. But let me take this privilege to first invite the Chief Operating Officer of Association of Biotech Led Enterprises, Mr. Suresh, and let me talk a bit about Association of Biotech Led Enterprises. They have been a pivot of promoting biosciences and they've been working tirelessly for past 20 years now towards the biotech sector. And you can see the result. You can see biotech sector transitioning from a $10 billion industry to a 80 billion, or very soon to be $100 billion industry. So let me take this opportunity to welcome Dr. Suresh to the screen, please. Namaskar, Dr. Suresh. Thank you so much for joining us today. A few words of wisdom from your side, sir.
Yeah, can you put up my presentation? I can just run through it.
Sure.
Uh, Thank you, Shekhar. Thank you, BioTecNika, for the opportunity to talk about ABLE and what we as an association have been doing to promote biotechnology. As you rightly said, we are founded almost 20 years ago in 2003, and this is our next year. We'll be celebrating 20th year. When we started in 20, it was a very, very small industry. I'll just take you through a few slides on where the industry stood last year. Because last 2 years have been very, uh, what you would say, very pivotal in the industry sector, because while many sectors showed negative growth, even national economy itself showed a negative numbers, this industry withstood all that because we are providing all the essential services and products for the um, pandemic. So you'll see from the numbers. So 2021 was a remarkable year. Almost on a daily basis, the nation added 220 billion to the national economy, and of course, our startups were all very vibrant and active during 2021. On an average, three startups were incorporated last year, and in all, 1,200 startups. And more than that, of course, all of us have had Covishield or Covaxin and many other host of vaccine. We are the only nation which has at least half a dozen COVID vaccine manufactured and supplied from within the country. We are not dependent on imports. On an average, we administered 4 million doses a day, and soon the nation's going to cross the 2 billion or 200 crore vaccine marks. And of course, R&D is also a lot of companies have spent a lot, and last year the industry's R&D investment crossed a billion. A lot of it went into COVID and related sectors. So, as Suman also said, it's one of the few industry which grew by 14% from 70 to 80 billion last year. This is what the Prime Minister referred to when he said in the last 8 years, Indian bioeconomy has grown 8 times. As you can see, biotech is not just one field. There are whole lot segments which make up this industry, and almost everyone has contributed. When some of the other sectors are not doing well, the COVID related products save the industry with substantial revenues. And also, foreign investment came in substantially in 2021 and almost 830 million. It's about 50 million more than 2020. And you may be, since there'll be a lot of students and others attending, so this industry is still primarily the biopharma sector, which is the vaccine, therapeutics, diagnostics, and others. Almost 53% of the industry. Rest is, of course, last year was different in the sense just the COVID and related RT-PCR tests, vaccines, and all others contributed almost 18% of the national bioeconomy. Uh, this may not be the trend later. Some other things will come in to replace, hopefully, in the next few years. Of course, biofuels, bioagriculture, BD cotton, and all doing well. And startups, as I mentioned earlier, startups activity was frenetic even during last year. More than 5,300 startups exist in the country, compared to about 50,000 across all technology sectors nationally. And so, just to let everyone know what we do at Able, we do a lot of initiatives for students. One of our most flagship initiative in the last 10 years is a Voice for BT public speaking contest on and through which supported by Novozymes, and we do it across four zones and the national finals, and the winners get cash prizes and also get internship in Novozymes and other top biotech companies. And this is done across various institutions. Since last 2 years, we have started monthly webinars. With the most recent one we did on how to commercialize stem cells on Friday. So various, almost all the topics in the industry are covered through these monthly webinars on the last Friday of every month. This is another flagship event started today, along with Women in Biotech Park in Chennai, of which Dr. Murli is a chairman. He will talk about it. So we have been recognizing the outstanding women entrepreneurs for the last 2 years. Then, of course, other job is to apprise the industry situation to the policymakers and regulators, which we do. We had the opportunity to meet and represent to Finance Minister Nirmala Sitharaman, Commerce Minister Piyush Goyal, and most recently with the DBT Secretary Dr. Rajesh Gokhale and other officials. And this is an ongoing process. That's one of the primary purpose of ABLE. And of course, ABLE enables many other events, including the Biotech track of the Bengaluru Tech Summit. The 25th year edition will be celebrated soon in November. And of course, Smart Bio Awards. And we collaborate with many international events, India, Bio USA, Bio Asia, Bio Taiwan. And we also bring out a India bioeconomy report, which captures all this data so that policymakers can rely on it, and so that they can plan to help achieve the national target of 150 billion by 2020, which is the industry has to double in the next 3-4 years. That's a very, very tough task the industry has. And of course, we are hopeful that there are many, many new sectors are coming up. Biological manufacturing, vaccines. Now that monkeypox or anything else the world throws at us, our companies and will be ready with all the products and services they need. Biofuels are catching up with almost 10% blending in petrol, sought to be which is targeted to be double to 20% by 2025. New sector of smart proteins, which are the alternatives to meat products, and new technical textile, biostimulants, and whatnot. There is a whole plate full of products from biotech industry. Able is at the center of this activity, or we would like to help everyone, because as this industry is not just one segment, and we would like to enable India achieve this dream of 150 billion by 2025. And you're all part of it. Many of you are members. This microbiome project itself, when it's around 2020, Dr. Yogesh Chauche came and talked to all of us in the industry in 2019 as part of Bioeconomy Conclave. We'll hear more about it. Back to you, Shekhar and BioTecNika team.
Thank you so much, sir. Thank you for your inputs. This really is some great work being done by ABLE. And my dear audience, as you can see, we are no longer going to be termed as a backyard of vaccine manufacturing. India is going to lead the world in terms of biotech sector, and Association of Biotech Led Enterprises and companies associated with it are going to make that happen. So with these thoughts, now we would like to have our event started. So our first speaker of the day is Dr. P.M. Murli. He's the keynote speaker of the day. And let me tell you about Dr. Murli. He's the president of Association of Biotech Led Enterprises Business Council, and he's the founder chairman of Genome Private Limited, Bengaluru. He has been a serial entrepreneur actually for more than 2 decades, and with many of first of its kind products and innovations in the areas of biotechnology. He has done some pioneering work in the IT greenfield projects, and in fact, he was a part of venture capital organization to evaluate prospective technologies for commercialization. So he has worked a lot into the biotech sector. Now, in his distinguished career, he has nurtured and built the entire Indian arm of 100 million Swiss biotech company. That is India's first pioneering public-private partnership to set up country's first genetic chemistry laboratory inside a premier campus, Indian Institute of Chemical Technology. So you can see, guys, he's a great renowned person of the biotech sector, and he's also built the R&D subsidiary of $250 million Indian FMCG group, Pioneer, in the world to conduct full-fledged clinical trials, all the 4 phases with polyherbal formulations for 7 disease indications over 2 decades. He's also known for his philanthropic and charity initiatives and is a great promoter of women in the biotech sector. So I would like to invite Dr. Murli. Over to you, sir. Thank you so much for joining us.
Thank you very much, the team BioTecNika as well as ABLE. So I'm humbled with the introduction that was given there. I think if you serve long enough, 30 years or 35 years in science, you do pick up a few things which are memorable. But first of all, let's get to today's topic. This is one of the most fascinating topic. I did start talking about it several years ago in Bangalore. And since then, I have been following this space and what's happening in India and abroad in this particular area. Now, for the uninitiated, the excitement of the microbiome research is as good as the excitement that you will have in exploring deep space, deep oceans, because both of them come with a lot of, let's say, darkness, mystery, a lot of things that you will not expect to find will be there. And a lot of things which you don't know is playing a big role in your life is actually sitting in these places. So our microbiome is also one such mysterious place, or a set of microbes control our body, our thinking, our metabolism, our life as a whole, that takes us to be a human being, where about one part is us, the Murli that is talking to you, and the 9 parts is hidden inside, where it is commanding me to talk what this one part wants to talk. So which means basically I'm completely being controlled by this. So it's almost like an alien story, right? It's so fascinating and so good and so great. So I will, before I go into the discussion on what I am doing, it's always nice to have a perspective, because in India, you would like to see the people who are at the policy making like to have a perspective of this industry. This has been very gradually growing, around 400 million, and primarily, in terms of diagnostic, therapeutics, all the type of interventions. But the beauty is it is expected to grow much, much faster in the next 24 to 36 months to almost, reach the 2 billion target. Now, why is that going to happen is because of a number of exciting things that the other speakers are going to share, and how this particular area is actually buzzing and it is planning to move to the next stage. But we will have to remember, I always keep saying this sitting out of India, is that we cannot be watching, we have to be participatory in anything that happens. So if you see that scale of investment that a country like United States is making, almost about $1 billion, vis-a-vis the rest of the world together contributing about 0.7 billion. And I don't know what share is India, but I'm guessing it must be very small. We need to be sure that we are upping our resources for this particular area, and we should be in a position to bring in a lot of research efforts so that we understand ourselves better. So that's the theme with which I think we should be moving on. So if you go on to the next slide here, you will have to remember that a bulk of the research so far has been done, and I would like the speakers to also elaborate more on this, is that a lot of the effort has gone in basically cataloging. Oh my God, what is inside us? So we are trying to catalog it. Oh, here is somebody, here is this, oh, here is that. Oh, these guys are a group of people. Oh, these people are together. So the cataloging has been going on for quite some time, and that thanks to a number of sequencing efforts, a number of ways in which we could actually look at things, we have now managed to come to a stage where we have done quite a number of cataloging. But however, we will have to now move from that stage of knowing who are the people involved, meaning which are the classes of microbes that are involved, to the type of relationship that they bring to the table, meaning how do they talk to themselves, how do they talk to us, how do we all talk to each other, and how is that a very big engine like the human self is actually moving with all these people who are visible and invisible talking to each other and moving this human body forward. Now, this means that we will have to start understanding relationships much faster. And even one small relationship that we understood, that we can have fiber, and the fiber will contribute to different type of acids, and those acids will signal something, and hey, here is a new set of nutritional products that can come out. So such is the type of possibilities and probabilities that come. So these short chain fatty acids, as they are called, is an outcome of the studies that have been done to understand how diet can regulate the way our body is behaving and what sort of metabolism that our body is capable of doing. So these are all some of the absolutely fascinating areas that are going to come into play. And we will have to remember even this title that we gave some time back about this being a mystery organ will fade away, as you all know, because organ is supposed to be more a static form, whereas with these bugs, and that too in trillions of them, mutating, moving, understanding the environment and changing. So it's a big cosmos of dynamic movement. So we may even outgrow these definitions of it being an organ. For the time being, we can park it as a mystery organ just because we want to put them under one bracket. But you know very well that they will not even fit into that definition anymore, as you will have to be sure that there's such a great dynamism that is there. So having set this tone, I would like to talk more about what I do in about 3 to 5 minutes, because I'm sure the other speakers have other things to talk about. So I'm in this exciting space called live biotherapeutics. So even the word means a lot. Live biotherapeutics means that you're trying to have a biotherapeutic, a factory or a medical store down deep inside you, and that will keep delivering drugs from bugs and look after the human body. So since I could be in this area for about 35 years, I could do bolder things that other startups may not be able to do. Almost on the border of being, being extremely audacious. So what we in our group felt is we should do something that is very, very challenging and that can have a path-breaking effect in the years to come. So what did we do? We decided that we will engineer a commensal or a bug that is already present in the human body. We will engineer a biosynthetic pathway. What does that biosynthetic pathway basically have? It can make a small molecule. And what is that small molecule? It's an anti-cancer drug. And what is this anti-cancer drug? This has been already used and it is approved by the FDA. All that we are trying to do is, can we make this molecule come out of a bug, which means that we will use synthetic biology to engineer it. So we have done that bit. Now, once you do that, you are almost saying that, hey, my payload is ready. So which means I can go out and deliver this payload to the place where I want to. But the most important part is there has to be an intelligent vehicle that will take the payload, deliver it when it is there, when it is needed, and it will shut up when things are not supposed to be done. So which means you basically have to make a very native commensal much more intelligent so that it is able to sense, it is able to deliver, it is able to do a lot of things that otherwise it cannot deliver. Now that's where cutting edge synthetic biology comes in. So a lot of work that we did in version 2, version 1 of synthetic biology, when we go to the next generation of synthetic biology, it obviously means that we are trying to do a lot more things, including the way we build circuits, the way we build intelligence into bugs. Now, this is one area that's been going on in the US. Fortunately, I have been doing a lot of work sitting out of India with my team here in collaborating with a number of companies outside India where they want to build this type of intelligent bugs. So I said, why not build one for myself? And I decided to look at one area, and that area is the respiratory system. So in the respiratory system, after COVID, everybody is aware what it can do to our respiratory system, what it cannot do, and so on and so forth. But the most important thing that we hear even today is that cancer, cancer, and cancer, especially solid tumors, especially small cell lung type of cancers, where the prognosis is pretty bad. There is no way in which a patient is going to live for too long unless and otherwise there's an intervention that is done. And more importantly, the intervention is to be done when all other types of interventions and opportunities are exhausted by the patient. So this is where we said, why not we try to engineer a bug in our company called eLive Bio, which is based out of US, but a lot of work is done in India, to see whether we can have a payload, make the bug intelligent. Can we formulate it in such a way that when it goes into the respiratory tract, that there is no inflammation or there is no trigger of immune systems, where it can go sneak in under the radar, seek the tumor, go and live next to the tumor and intelligently deliver the drug that is being put inside the bug, and see whether it can prevent a lot of collateral damage that the patient will otherwise have, and can it also help in his quality of life. So this is the area that we have been working for the past 2 to 3 years. I have to say this is really where it separates men and boys. And sorry for saying this, because boys don't want to get into this area because it's a hell a lot of risky, a hell a lot of challenges are there. But the Star Trek mission is the same thing that some of us have in science: go where no man has ever gone before. And that's how I have been leading my life for the past 30 years or 3 decades. A lot of the people in this forum know. So I would like to end my talk by saying I am extremely excited that I'm going into deep space once again, completely unknown, blindfolded, but a group of people also coming along with me. It is one of the most exciting space, and if we are able to contribute something in this area, we will be delighted. And so I would like to end this talk by mentioning that this is going to be a new frontier, a great area to work, and you can see palpable excitement when I talk about my science, and you're going to be seeing the same with the other speakers as well. It has been such a blessing to come out and talk to you about what I am doing today. And I would like to thank BioTecNika, thank Bala, Suresh of Able, and I wish this seminar, webinar, all the best, and hope that I come with much more news when I meet you on the Microbiome Day next year. Thank you very, very much.
Thank you. Thank you so much, Dr. Murli sir. It was really insightful. And wow, the work you're doing at eLive Sciences, it's brilliant. And the approach, definitely, it separates the men from the boys. I wish you all the best. And definitely, we look forward to the next World Microbiome Day celebration, when you will have some great success to share with all of us. Wonderful approach there, sir. And I'm myself very much intrigued by listening to this approach. Trust me, wonderful. That's like, let's create a super bug which can just go in and bug the cancer. Wow. It's a super insightful approach, and brilliant. Thank you so much for sharing this with all of us, sir. So now, guys, now, my dear audience, we have our second speaker for the day, Dr. Rekha. And she is the professor and director at Yenapoya Research Center, Yenapoya University, Bangalore. She has a PhD degree from the Department of Biosciences, Mangalore University, in the year of 2002. And Dr. Rekha continued her research as a postdoctoral fellow at the National Chung Hsing University, Taiwan. till 2008. And after that, she joined Yenapoya Research Center in 2009. And from that time, she has played a significant role in the establishment of multidisciplinary research center at Yenapoya University. Her research interest focuses on biotechnology, applied biotechnology, microbiology, biomaterial research, and she's with us today. So I'll quickly invite Dr. Rekha to the screen. Namaskar, and thank you so much for joining us, ma'am. A few words of wisdom from your side on this grand celebration day. Over to you, ma'am.
Yeah, thanks for that. Yeah. And it's a great honor to be here with among all the people who are like leading the way for Indian biotechnology sector. And every scientist's contribution for this is going to make a great deal for the national development, particularly in the field of biotechnology. So right from my early days in Chongqing, like biotechnology was a fascinating word. So we were working mostly on the microbial diversity and various sectors. And coming back to India, like in a medical setup, my research focus looked into the microbial aspects in diseases and health. Yeah. So as you all know, today is the World Microbiome Day. And at the end of the day, like all the listeners definitely would have got a lot of good insights into the microbiome. And I'm definite that everyone is quite familiar with the role of microbiome, particularly in the health, environment, agriculture, and where not, say, as it is like a mysterious word, we know many unknowns. We know that there are many unknowns than knowns in this microbiome world. So with that, I'm going to just talk a little bit about my research. Just starting before that, I will just tell you a little bit brief about the microbiome, like how it colonizes our system, what is the important cooperation is needed from our body or any environment particularly. So as all of us know that we are one among million genomes in our body. Like if suppose a human organ is a human has been treated as a species, we are harboring several thousands or millions of the microbes with us. Let it be, it can be virus, it can be fungi, or it can be bacteria or archaea, all of them together with us. They live in a healthy cooperation. Sometimes they may be not that happy with us, like, so they will play a little bit tricks and may end up with some diseases also, like some conditions, pathologic conditions called infections. So we all know that we are born and live in an environment full of life. So if you go back and see how life evolved in the micro scale. So we know there are several millions of life among us. So as a baby, newborn baby born in either the normal route or through a C-section in a sterile measure in an OT section in an operational theater setup, but immediately afterwards we are transferred from one hand to the another hand, right? And then air breezes and we are washed or bathed in water. So there are everywhere there is a life in contact with us. So it's kind of a cradle made up of microbes for us. And we grow, like we may have a handful of friends for us, but you consider each of the tiny microorganism as your friend. So you have millions of them to you. So there is no wonder the life evolves or we grow as the milestones of growth in the chart. But our number of friends, the microbial friends, also increase day by day, and we become more diverse and with multiple species with us. And then if we see that, like, are we so like disposed to these microorganisms? Definitely not. So for a successful colonization, successful friendship for any microbes, like let it be fungi, bacteria, or virus, there should be an understanding between the host system as well as the microbe. So there should be a cooperation, and then in the microbiology we say that's the host microbe interaction. This is not only with us, but it is also with the plant or environment or any organisms. That's must for any microbe to colonize, there should be an understanding and cooperation called host microbial interactions. And this is what, like, when we exchange a handshake between friends or anybody, or we exchange a hug between an animal and us, there is a lot of transfer of the microbes from one to the other. So when I have talked about like where and all we live and where and all we have microbes that can get incorporated into our body, like say from skin microbiome to gut microbiome to lung microbiome to nasal cavity microbiome to an middle ear microbiome. So we have many microbiomes in us, like the biomes in us. Biome is ecologically is a big term and has a significant value also particularly. So we have several biomes within us. So we harbor several organisms in different niches of our body. So urine microbiome is such one mystery organ within us. So let us see what is urine microbiome. So if you have seen the human microbiome project, there are several the different niches, body parts have been used for the human microbiome project. But unfortunately, fortunately, or knowingly or unknowingly, the urine or the bladder was missed out. Why? There is a story also for that. Like for decades, urine has been considered to be sterile unless there is a pathological condition. If at all there is any organism in the urine, that is to be considered to be an infection. But subsequent studies after the human microbiome project, the studies on the urine, several studies have independently taken up that identified the healthy urine also contains several microorganisms. So then became a new term called urinary microbiome or the urobiome. So our urinary system consists of, like all of us know, that kidney, bladder, and ureter in between, and the urethra, and then finally it is excreted, the urine is excreted. And if you see the anatomical structure, the urinary tract is in close proximity with the gastrointestinal tract. So how can you expect the microbes do not colonize the urinary tract? There is no mystery behind that. Like that is quite obvious that we can have a urine microbiome or the uromicrobiome. Urobiome. So now, if at all by definition, if you want to see what is a urobiome, that is the microbial community in the bladder obtained urine. So the techniques what is used for collecting the urine may be different, but generally it is defined as the bladder obtained urine, the microbial community in that. And as like other organs have, the microbial diversity is not yet known. Even for the urine, also the whole microbial community is not yet well characterized or understood. And it is more abundant. In terms of urine microbiota compared to the gut, the urine microbiota is less abundant than and less diverse than the gut. For example, the female urinary microbiota may contain up to 10 raised to CFU, that is a colony forming unit per ml. And if you compare the same individuals, the feces, you may have the double or more than double of the bacteria, double of the microbial load in the feces that is coming from the gut. So this is the difference right now. This may be due to the sampling errors, or this may be also due to the difficulty for the microorganism to colonize the urinary tract. And if at all, based on the urinary microbiome studies, people have already made different urotypes, identified different urotypes. Like the enterotypes for the gut microbiome, people have identified, scientists have identified. There are urotypes that have been identified from across the studies. There are 10 urotypes, for example, between the female and male urinary tract. Because of anatomical variations, you have significant differences in the urinary microbiome. For example, in a study, like from a couple of studies, people could identify 10 different urotypes from a female urinary tract, while from the male, this is the healthy males, they could identify only 6 urotypes. So the diversity between the two sexes also have been seen. So if at all you can identify what are the major urotypes, there are some are common, which I have like you could see in pink color, that is common ones, like example, like the Prevotella amni, and then Shigella soneri, etc., and Enterococcus faecalis, etc., are quite common. But there are also a few others that are unique to the male or to the female the urinary tract. Another thing, the most important thing we have to know that the urinary tract, as I said, is a little bit difficult for the microorganism for the successful colonization, because there are more than 2,500 urinary metabolites that have been identified. These metabolites may be you know that the urea, creatinine, and many other excretory products that come out in the way. And it is also kind of a salt-rich environment for the organisms to colonize. So there are a lot of difficulties for the microorganisms to colonize successfully, because it should have such an adaptations to colonize. So if you also look into how we can acquire a urinary microbiome, there are ways in which we can always theorize, or we can understand that how the urinary microbiome is built or established. One is that its close proximity to the gastrointestinal tract, that is colonized from the oral cavity during the birth to the your alimentary canal and to the anal, this one, and then it can also have a migration of the microorganisms to the genitourinary tract. So that is quite easy. If you can see a small baby from the diaper days, how you can have the contamination of the genitourinary tract and colonization of the microorganisms. And whatever the microorganisms, they can also form the ascending colonization in the urinary tract, because they have the physiological and metabolic adaptations, particularly the structural adaptations like pili, fimbriae, and motility allows the microbes in the urinary tract that can successfully colonize to ascend the upper part, like into the bladder, and sometimes even into the kidneys. And then they also have high metabolic adaptations. We know from several genomic studies that microbes have diverse enzyme systems and metabolic capabilities. One of the important one, because there is a lot of urea, the excretory product is in the urine, so there should be urolytic enzymes. So many of the organisms do have enzymes called ureases, but not always, because if we see that less than 10% of the microbes in the urinary tract do have urease enzymes. But there is also alternative urolytic enzymes like urea carboxylases and several others that are still being studied. And there are different techniques where you can have culture-based and culture-independent techniques, but in the microbiome studies, the culture-independent techniques are mostly valued because culture-based techniques are limited. But I may say anybody who is doing research in the microbiome studies, just don't go only onto the culture-independent metagenomic approaches. Kindly also look into the culturable bacteria or the culturable organisms, because you can learn a lot more. I will just give an example like how we can learn a lot more from culture-based while you have the metagenomic approaches also. And this is a case study what I'm going to talk to you today, because this is from our experience. Like, we conducted a study on the kidney, the urinary tract microbiome, particularly uromicrobiome, and we took two groups of the subjects in these like conditions. One is healthy group, another one is the kidney stone disease. Why we have studied this? This was in 2014 we started this project, because by then that urinary microbiome was just getting evolved. Why we were seeing? Because we were seeing a lot of kidney stone patients in our hospital that is attached, and we were seeing what is the role of different microorganisms in the kidney stone disease. So what we did, we just collected the kidney stone group patients from the kidney stone group, and we characterized the different types of kidney stone, and we identified what are the different major types of kidney stone. We could identify 12 major types of the kidney stone group in this study. And among that, we found that there are one group particularly going into calcium oxalate group. You know, there is a gut bacteria that is oxalate, oxalobacter, that can degrade oxalate and prevent from the kidney oxalate stone formation, but it's a gut bacteria, not from a urinary tract. Whilst dry stone, everybody knows it's a stone, infection stone we also call it, because of the urease positive microorganisms it is caused. And there is uric acid stone and mixed type of stones. So we compared the differences between the microbiota or the major genera between these different groups. And as you see in the screen, you can find out that there is a dysbiosis in the UMB of the kidney stone disease compared to the healthy stone. So where you can see some of the major groups that is abundant, particularly in the healthy, the Prevotella was predominant. That is the patients from the southern India. While in the KSD group, the whole of the KSD group, the kidney stone group, you could find more Enterococcus species. While in a particular stone type, there's a calcium oxalate, you will have the Staphylococcus. While in the uric acid, the Prevotella was highest, but the second one was the Enterococcus. While in the healthy, the second most predominant genera was the Lactobacillus. So you could see a lot of dysbiosis. So we also could associate 30 genus into the different stone forms. Why this is important for us to study was that what protects an individual, what is a healthy microbiota that we can manipulate to prevent kidney stone disease. Because once a patient gets a kidney stone from a particular type of kidney stone, he or she is prone to have recurrent kidney stone disease. But recurrent kidney stone disease can damage significant amount of the kidney as an organ, and that is going to cause a lot of health problems subsequently, because it's one of the major organs present in the human body. So that is where we were particularly looking into. And interestingly, we could find two important things. There were one Lactobacillus strain. There are two Lactobacillus. Actually, there's a Lactobacillus planturum. Two strains could protect the patients from the oxalate stone formation. That's a different study that we have conducted. While another interesting thing, what we have identified in that is what I said, the culture-based approaches were important. We could identify an organism that was not yet identified yet, like it was a new species and it was mostly a new genus. So we were just holding on from where this new genus had come to the urine of a patient who was having a struoid stone. And subsequently, we did the whole genome analysis of the same organism, and we were just analyzing particularly with the interest its role in the kidney stone disease. When we were doing that, simultaneously there was a study, an interesting study. The same isolate was identified and described as a new genus from the International Space Station, from a group from United States of America. And that shows that the metagenome to phenome, an approach enables isolation and genomic characterization of Calamiella personi, genus novel and species novel from the International Space Station. By then, we were in the midway of our work. So we had already identified, and that study shows that it has antibiotic resistant genes, but it may not be human pathogen, because they did a software called Pathogen Finder, and in that it shows the pathogenic percentage, but it was not showing pathogenic trait. But while we had isolated this organism from a patient, we were looking into the role in the struoid stone formation. So we were doing a lot of studies on that. But it was urease negative. It was surprising for us that it's a struoid stone associated microorganism and it's a urease negative. And how does it implicate? Does it really have a role in the struoid stone formation? So we did a lot of in vitro studies, and we also did the whole genome studies for that. And we identified that this bacteria could produce ammonia that is needed for the struoid stone crystallization. And that ammonia was produced from the amino acid degradation because of an enzyme called asparaginase. And we did in vitro tests, like by growing it in the synthetic urine, it could grow, and it had several virulence factors that are capable of colonizing the urinary tract. And it also formed some amount of small crystals that was identified by machineries like FTR and then XRD to be identified as struoid crystals. So that gave us one evidence that it has a role in struoid crystallization. And subsequently there were a couple of reports quite recently, and when we are currently working on its alternate urea degradation pathway, that is the urea carboxylates and aliphatinate hydrolase, and simultaneously now we are ready with the manuscript and other things, because we want to communicate this to the people, because those who are in the health sector, particularly those who are looking into the infectious disease, please do not ignore this pathogen because it may be significant. So, and when we were doing this work, we found out that there is another publication that is coming from the New York, from the Rochester University, where there is a case of human disease caused from Calamella piersoni. So what we claimed it as to be a pathogen has turned out to be true. That was why the culture-based approach was also parallel approach could establish some amount of links between human disease and this organism. And there is also those people who are working on taxonomy and evolutionary microbiology can also be interested that this bacteria was later put back into Erwiniaceae to the Pentoia group. Right now it has been reclassified as Pentoia piersoni, and we are studying on its role in the alternate urolytic enzyme system because it harbors a urea carboxylase and aliphatinate system that can break down urea successfully. So now what is the scope for the study? Because when unknowns are there and it may have implication in the disease, so you can identify what kind of antibiotic treatment can be given. And these kind of studies also explore the unknowns, and we can identify more determinants of the disease where the preventive measures can be taken, or we can also have some like the antibiotic therapies, etc., or the targeted therapy specifically rather than giving a broad range of antibiotics. And coming back to the study where our research center is there are in our group there are 5 of us. All of us work on different areas of microbes, but all of us do have work on the microbiome related studies for various. While I do work on urine microbiome in kidney stone disease, one of my fellow colleague, Dr. Arun, works on gut microbiome, works on the oral microbiome in cancer and its treatment, how the colonization disappears during the treatment and how we can facilitate recolonization. And as a part of the study, we are also studying the oral microbiota and its role in the oral cancers, where we are looking for the organisms that can protect the patients from the oral cancers and to be treated, to give them as prebiotics or the probiotics actually for the high risk group. And there is also the gut microbiome and its crosstalk in the colon cancer by Dr. Rajesh Shastri, who works, and that is for targeted therapies. And similarly in the environment and resistomes in the aquatic systems by Dr. Asif and Dr. Bhagwan. So with that, I just want to conclude the session by saying thank you to everybody, the organizers like the Able Foundation, BioTecNika, and everyone. So maintain your urobiome and have a healthy urinary tract so you can prevent a lot of diseases. And there is also a lot of scope for those people who are working on different the niches, human body niches on the microbiome to develop therapeutics, targeted therapeutics, etc. So let's all work towards one cause, that is for one health. So healthy microbiome in the human body also is a healthy environment. So with that, thank you once again, everybody. Thanks for this very good opportunity to share our research and for the showcase our research activities. Thank you.
Thank you, ma'am. Thank you, Dr. Rekha. So quickly, guys, as we are short of time now, so we'll quickly bring in Dr. Gautam Das. Dr. Das is the co-founder and managing partner at MyBiome Therapeutics LLP and chief scientific advisor at Microbiome Research Private Limited, Mumbai. He is a scientist with 20 plus years of research experience in life sciences and diverse experience in genetics, molecular biology, biochemistry, genomics, cancer biology, and cell biology. He has handled numerous R&D projects and commercial projects and managed large group of people. And today he's going to summarize all his accomplishments through this session with you all. So over to you, Dr. Gautam. Thank you so much for joining in. Namaskar. And you can start.
Thank you, Shekhar. Can you see my slides? Hello, everyone. Yeah. Hello, everyone. First of all, happy World Microbiome Day from all of us here. Today, I'll be talking very briefly about our work on the role of genome, as we all know, and second genome, which is the microbiome, which is the focus of today as we celebrate World Microbiome Day, in human health, wellness, and disease from all of our efforts at MyBiome Therapeutics and Microbiome Research. So Special thanks first to Bala, who first invited me to this talk by Abel, and then BioTecNika came in. So thank you to all the organizers. So let's start with a poem. I write poetry, so let's start with one of the poems that I recently wrote. Tiny trillions inside us, who is powerful, they or us? We humans think we got guts. The invisibles think they are the guts. We believe our master is brain, but gut is called the second brain. We all are born with our genome. Did you know the second genome? Time to pause and ponder today while we celebrate World Microbiome Day. When mighty trillions shape us better, 3 billion bases perform better. So who are we? MyBiome Therapeutics and Microbiome Research. As I said, we are a fully equipped advanced science and deep tech startup company in Mumbai. We have a state-of-the-art lab here in Mumbai where I'm sitting now. We specialize in microbiome research, diagnostics, genome sequencing, genome-based diagnostics, bioinformatics, genomics research services. We are backed by promoters of Excel Group, who have a vintage of more than 80 years now. We have set up a laboratory in 2020, so less than 2 year old, with Illumina and Oxford Nanopore and all other molecular biology infrastructure. I'm very happy to share here that in a very short span of time, this company has generated more than 3 trillion bases of genomic data, which is a yardstick of any genomic measurements, processed over 1,500 genome samples, and delivered over 100 research projects for clients. And this majority of them include microbiome research projects from different reputed government and private organizations. We have a team of now 20 plus experts in the field of NGS, molecular biology, computation biology, microbiology, software, and business. This is our team, full of energetic scientists, engineers, business professionals, and here is me and my co-founder Ravi Shroff here. Our core areas of research started with a simple vision that genomics should be for all, and genomics for all is our vision. Our core areas of research, apart from microbiome, which is the focus of today's celebrations all over, are into infectious diseases, where we focus on bacterial, fungal, viral. We moved into COVID research, and then HPV. We focus a lot on drug resistance, tuberculosis drug resistance, Klebsiella pneumoniae. We focus a lot on hospital microbiome, which is very important in hospital-acquired infections. And then another part of the company also focuses on cancer, reproductive health, inherited diseases, as we are a genomics lab. And recently, we have been also focusing a lot on immune typing, that is HLA typing. Very happy to share that we have been very active in research, and we have been publishing book chapters, research publications. We have been patenting our research, intellectual property, reviews, preprints all over. These are our research services and clinical services. Our clinical services mainly focus on infectious disease, as I said, cancer, genetic disorders, HLA typing. Our research services are all over into DNA, RNA, metagenomics, epigenomics, and computation. Our clients include clinicians, diagnostics, hospitals, academic research, pharma companies, and the government. So to start with, what we do in our company is simple. We try to decipher the role of genome in precision medicine, and we call it MyClinical Genomics Research and Diagnostics, MyClinGen RX, where we receive samples, we process the samples, we analyze the data, we do reporting for clinical and as well as research, and we store the data for further research using Indian data, which is a very rich source of data, particularly for our population. So this is how MyClinGen RX works. We integrate Indian data every time we get more and more Indian data. So this is our data analysis pipeline, exome, genome sequencing, etc. And then finally, it goes to consumer and clinical reporting. We have been recently doing a lot on Indian genome-based cholesterol panels, exome analysis, HLA typing, and pharmacogenomics. Now moving on to what we do in the second genome, which is the role of second genome, or microbiome as we call it, in precision medicine. And for this, we have a specialized entity called Microbiome Research. As you all know, gut microbiome is a very special area in the area of microbiome. It is made up of trillions of bacteria, viruses, fungi, archaea, protozoa, and I know Kumar is here today, and Vinod, who will be talking a lot more on this. To understand what makes a good gut, researchers are investigating the gut microbiome in vast, vast detail. Genetics, lifestyle, diet, geography, age, everything can affect our microbiome. So I wouldn't go into the details of what we do research into the gut microbiome, but this slide summarizes everything that gut microbiome can affect inflammatory bowel disease, obesity, diabetes, cancer, infectious disease, psychological disease, mental health, autoimmune disease, and everything else. So here is what we do. Our focus is a journey to a healthy, happy gut. So we use all those research studies from genetic and functional characterization, in vivo studies, ex vivo studies, in vitro studies, microbiome data from all over the world, model studies in mice and rats, clinical studies from humans, and then bundle it into a personalized recommendations for prebiotics, diet, probiotics, and then we give the customer or the patient recommendations through which he or she can restore his or her gut. We use this information in our research, and this is particularly being used for chronic disease management like IBD, etc. Now, a few lines about what we are doing right now. To summarize, I feel a gut feel, feel I. If you feel the feel I feel, you will take the test as I. And I welcome you all to take this test which is a journey to a healthy, happy gut. It has been made very easy through the click of a button at www.microbiome.in. And the costs are very, very attractive. So please take this test. Now, beyond our gut, we have a lot more in the space of microbiome. And just now we have heard the earlier speakers, Dr. Rekha, Dr. Murli, speak a lot on other microbiome areas. So we have been also focusing on nasal microbiome, oral microbiome, skin, lung, urinogenital, etc. And because dysbiosis of any of these microbiome could lead to many, many diseases. And this, how do we analyze the second genome in our company? We have workflows for metagenomics pipeline, which uses different bioinformatic tools and statistical analysis. We also use different statistical tools and visualization methods to analyze the second genome or the microbiome. The R&D projects in our company, as I said, we focus a lot on gut microbiome and diet. Recently, we have been doing gut microbiome and IBD, gut microbiome and COVID. We are also planning a study on gut microbiome and diabetes. And then we also did a study on vaginal microbiome and pregnancy and respiratory microbiome. Now, to move from our own DNA to our second genome, which is the microbiome, we cannot ignore the external agents. And that is why in our company, we focus a lot on infectious diseases. And within that domain of infectious diseases, we all know that tuberculosis is a global threat. However, we are making advancements in research in tuberculosis. Still, 10 million people fell ill with TB in 2019, and 1.5 million die from TB each year, even now. So in our company, we have made a TB targeted next generation sequencing solution, which is a very cost-effective solution. It is cheaper, better, and faster, as it works directly from the sputum of an individual. Now, moving on, in infectious diseases, we also focus a lot on human papillomavirus. Why do we focus on human papillomavirus? Because 6.6% of Indian population suffers from sexually transmitted infections. And within that, human papillomavirus is a major infectious element in sexually transmitted infections. And this causes cervical cancer, which is the second most common cancer in Indian women. So I wouldn't go into the details of all this, but what is important to note here that we are implementing a powerful tool for detecting multiple human papillomavirus and sexually transmitted infection types using next generation sequencing, as our company focuses entirely on next generation sequencing. Once we have the data, bioinformatics and genomics is very important. When technology meets science, science meets technology, I say magic happens. And this is what this slide represents, that when tech meets science, science meets tech, DNA sequencing can lead to further tools of AI, ML. And a famous mathematician, Clive Humby, once said, data is the new oil. And I am a firm believer of this, because the magnitude of data that is held in public, private, and commercial databases just with the DNA data is unprecedented. So AI and ML are promising to be breakthrough for medicine, and I encourage young students who are taking biology, or even young students who are taking computer engineering in their careers, to choose biology and genomics as their career, as this is where the big data is. Whatever we speak of e-commerce, but the magnitude of data that can come out of big data that can come out of human genome is unprecedented. So with this, I would like to end this talk here. Please feel free to reach us at hello@mybiome.com or info@microbiome.in. Thank you, everyone.
Yeah. Thank you so much, Dr. Gautam. It was really insightful. We thoroughly enjoyed the statements which you made there. Data is the new oil, like you said. And truly speaking, the NGS part and the genomics kept me intrigued throughout the presentation. Thank you so much for joining us today. And happy Microbiome Day to you too. So we'll now start with Mr. Kumar Sankaran. He is our next speaker. He's the CEO of Lucin Rich Bio Private Limited, Bangalore, and is a designated partner of Gene Express LLP. He's a business leader with a professional experience of 15 plus years in healthcare research and delivery. In his current role, he is spearheading the growth and innovation of microbiome domain for the Asian market. He is passionate about driving the paradigm shift from sick care to healthcare. So let me take this opportunity to invite Kumar Sankaran sir. Namaskar. Welcome to the session, sir. If you could have some thoughts shared with all of us today. You are muted, sir. You have to unmute it. Yeah.
Thank you, Shekhar. I hope you're able to see me and hear me. Hello, everyone. Happy Microbiome Day to one and all. I would like to first of all thank Bala from Able to inviting me to this webinar, as well as Able and BioTecNika for creating such wonderful session. My talk today would be slightly different. I wanted to take you all through the different vantage points from which you can view the human microbiome. And by viewing it differently, you'll be able to see how the microbiome is different and how it impacts us differently. I like to show this number that, some scientists say that there are about 10 times more microbes in our body than human cells. And one of the reasons why I like to show this number is to say that it's such a powerful group of cells coming together to actually define the way where our health is and where our wellness is and how it shapes our mood, our hunger, our energy, everything related to human body metabolism. So there are about broadly 4 different ways in which you can view the human microbiome. The previous speakers, especially Dr. Murali, was talking about how it's traditionally now being looked as a mystery organ or an organ, but that definition is slowly dying. We can also look at the human microbiome as an immune system, where all the microbes are in our body are actually preventing microbes from outside our body to come into our body. The third, the most interesting view of all is the holobiont or the whole genome theory, which is also a very exciting way to look at the human microbiome. And lastly, if we can zoom out and look at it from a satellite level point of view, you can see the microbiome as an ecosystem in which the humans are merely that play a very small part. So let's dig deeper into all these views. So when you're looking at the organ view, it's a very simplified way of defining the human microbiome. You're saying that it's a new mystery organ that has been discovered, and that needs to be profiled, cataloged, measured on a regular basis. Saying that this view does not hold up most of the times because the microbiome is constantly changing, and organs, seldom change, or if they change at a very, very slow pace. But the microbiome is constantly changing, and it is also not localized to a specific part of our body. It's present all over our body. I mean, we just now heard about the fascinating research that's been happening in the urinary microbiome. So there are different kinds of microbiome niches across our body, and that is interacting with each other and constantly changing. The second type of view is mostly looking at the microbiome as an immune system. So it is, like I mentioned, can the microbiome act as a protector against pathogens? We published a paper, one of my co-founders actually published a paper in 2020 where they reviewed the role of the gut microbiome in prevention of lung diseases, especially lung infectious diseases. So it kind of showed how the gut microbiome, using the crosstalks between the gut-lung axis, are able to defend new microbes from attacking the lung. So so looking at it as an immune system is a good way to look at it. Some scientists and researchers all over the world actually kind of call it the second immune system. The biggest question is, can the microbiome differentiate between self and non-self? Or is it mainly a group of cells trying to protect its own territory? So you have a certain kind of bacteria that's come and colonize the gut, and do they then fight against anything that come and want to colonize that region? Is it merely that, or do they also think as this human system as its own self, and something foreign to it as a non-self? So that's another vantage point from which you can look at the microbiome. But thirdly, and like I mentioned, I mean, for me, the most fascinating piece of it is the holobiont, or the whole hologenome theory, where human cells and microbial cells are acting with each other, they're evolving with each other, and they're being complementary to each other. Um, like Gautam Das in his earlier discussion was talking about the primary genome and the second genome. It's how these microbial genetics, how is that interacting with our human genetics, and making us healthier, or probably leading us towards a disease state. So the holobiont and the hologenome is a very, very new, interesting concept. The one of the drawbacks of this view is, there's a question mark to it, is whether it's an heritable system. We know the human genetics is inherited, and we get it both from both our parents. But is the microbiome also a heritable system? We know a lot of the early infant microbiome is actually got from the mother's womb and the birth canal. Also, a lot of the early microbiome is got from mother's milk, and how that milk is actually delivered. So in that sense, there's some kind of heritability in the system, but not as defined and as concrete as the human genetics is. So although this is a very, very good vantage point from where you can look at the microbiome, there are questions about whether this has hereditary properties, and by that can we dictate how our offsprings are going to be. Lastly, the ecosystem view, where you're zooming out. Now I'm sitting in this room, which is full of microbes interacting with the microbes on my skin, which internally is interacting with my gut microbiome. So looking at it as an ecosystem where you're looking at the habitat, you're looking at um, how different kinds of environments are actually interacting with the microbiome within us, and thereby improving health in that manner. I'll give you a very small example. We've done tons of consumer microbiome studies from all over India. With a fair degree of accuracy, I can predict whether a person is from a rural setting or from an urban setting. And that is because in an urban setting, you're interacting with that much more pollutants. You're getting pollutants from the air, from the food, from from the environment in which you exercise or sleep. All of that adds to it. There was also a recent study which shows that exercise independent of diet actually changes the gut microbiome. All right. But what was also very key to know in these studies is that where you exercise also matters. If you're running in a nice green park or running on the beach, that affects your gut microbiome differently to probably working out indoors and doing a treadmill within a gym. So all of that is interacting uh, with the different microbiomes in your body and changing it accordingly. So the ecosystem view is probably the holistic view of the microbiome, but where it adds to the complexity is in a way that there's no clear boundaries over here, and you don't know what exactly microbiome needs to be profiled. Are you just happy with profiling your gut microbiome, the skin microbiome, or do we go in and see your inbuilt microbiome or the microbiome of the food that you're eating? So all these kind of complexity and practicality comes into question. But the ecosystem view is probably the most comprehensive view of the microbiome. So in a summary, um, you sit at different vantage points and look at the human microbiome, and then you begin to put practical buckets to each individual view. Um, it is good to view it as an organ because then you know what you're expecting a certain outcome, and you know what a bad outcome. um, if you view it as an immune system, you understand what dysbiosis does to the immune system, what eubiosis, that is a good diverse microbiota, is actually doing to the immune system, and maybe just measuring diversity of a particular microbiome within our body is actually very good. If you look at it from a holobiont view, you're comparing your microbiome with your genomics. You're trying to see, because of your genetic makeup, what is a good microbiome you need to have, and vice versa, and see how these two things are interacting with each other, and you can focus on building uh, your microbiome to match your genetics. And the ecosystem view is managing the habitat. It's managing where you live, what kind of air you're breathing. It's not about looking at sterile and non-sterile condition. It's more about how much of chemicals are there in your environment and how that chemical is actually um, ruining both your ecosystem as well as your microbiota. I'll give you a very simple example. There was a study that was published in Nature about 2 years ago, which showed the difference in the apple microbiome. So there were two sets of apple. One was grown without the use of pesticides and chemicals. The other was grown with using pesticides and chemicals. And the end produce, the apple, which used pesticides, had very less diversity of, of microorganisms, whereas the apple, which was grown using no chemicals, had a very rich, diverse set of microorganisms, which then would benefit your gut microbiome. So the key point to remember over here is the diversity is actually pretty good. So, these were the different vantage points in which you view the microbiome. And we here at Lucent Rich Bio, what we've done is we've built a product called as Bug Speaks. By the name, what we're trying to imply is, there are microbes in your body, and they're speaking to you, and they're dictating a lot of your health and wellness. So Bugspeaks is a cutting edge, easy to use at home microbiome test. We've been working on this, right from 2014. We've done a lot of R&D using data from all over the world to see how the microbiome is actually affecting our body. Bugspeaks also detects susceptibility to about 16 diseases right now. So how does your microbiome translate to a person getting diabetes, obesity, things like depression, Parkinson's, the various kinds of non-communicable and chronic diseases that we're seeing all around us. So it detects susceptibility to about 16 diseases. We don't stop at the susceptibility part of it. We also provide nutritional recommendations to correct any kind of dysbiosis that is there in the gut. It provides prebiotic and probiotic recommendation. Bugspeaks has a couple of product. One is a couple of patents. One is a product patent, and the other one is an intestinal health marker, patent around the intestinal health marker. So we've taken a lot of effort in coming up with a very simple score to tell you how good your microbiome is. If it's towards a negative number, that means your microbiome is in a bad state. If it's towards a positive number, that means your microbiome is in a good and healthy state. And right now we're sitting on one of the largest Indian microbiome data. That's because we've been working on the microbiome, since about 2014, and we've collected data from all over South Asia, in fact. So with that, I would like to end my talk. I would, just mention one thing at the end of all of this is, take care of your microbiome, and it will take care of you. Stop disrupting it. Stop using harsh chemicals to eradicate your microbiome. Harsh chemicals like like antibiotics or very harsh chemicals that are going on your skin. Try and stop using that, and you'll have a healthy microbiome, and the healthy microbiome will actually help you stay healthy. Thank you.
Thank you, Dr. Kumar Sankaran. Thank you. That was really insightful, and wow, what a line. Respect your microbiome, and they're gonna respect you. Thank you so much for those insightful words. So next up, quickly, guys, we have our last speaker of the day, Dr. Vinod Kumar Gupta. He's microbiome program member at Mayo's Clinic Center for Individualized Medicine at Minnesota, US of A. Dr. Vinod Kumar Gupta is a postdoctoral researcher at Mayo Clinic, USA, and his doctoral research is focused on delineating taxonomic and functional divergences of microbiome components across different human body site niches. He's currently investigating on the role of the gut microbiome in the progression of rheumatoid arthritis, and he's also working towards a better understanding of the human gut virome and its relationship with human health and diseases. So I would like to quickly invite Dr. Vinod onto the screen. Thank you.
Hi everyone, my name is Vinod Gupta. I'm a postdoctoral research fellow at Mayo Clinic, and before I begin my talk, I would like to thank all the organizers, especially Dr. Gautam Das for inviting me to talk about my work. And it's a great pleasure for me to talk about the gut microbiome on the occasion of World Microbiome Day. And recently, I have been seeing that the gut microbiome has been emerging as a new frontier in the healthcare system. So, to understand the gut microbiome, how it works, let me begin with the definition of gut microbiome. So, collection of microbes live in the gastrointestinal tract, especially in the large intestine, that includes bacteria, viruses, protozoa, and fungi, and their genetic material, collectively known as gut microbiome. And when these gut microbes lives in a homeostasis with our body, they play an important roles to keep us healthy, such as synthesis of vitamins, production of short chain fatty acids, and training of our immune system. But any deviation in the gut microbiome composition from its normal or healthy gut microbiome composition may cause various diseases such as inflammatory bowel disease, type 2 diabetes, or colorectal cancer. And deviation includes loss of beneficial microbes or colonization of pathogens in our gut. And the deviation in the gut microbiome composition can be triggered by many external factors such as use of antibiotics, change in lifestyle, change in diet, and medication use. So, and recently many gut microbiome studies have found a huge variation in the gut microbiome composition among healthy individuals. So, and which makes the characterization of healthy gut microbiome composition very difficult because and indicate that this huge variation among healthy individuals in the gut microbiome composition indicate that there is no specific healthy gut microbiome composition. So there might be many healthy gut microbiome compositions. So so it's really difficult to measure the deviation in the gut microbiome composition from its normal or healthy gut microbiome composition. So to address this issue in our study, we aimed to develop an index which can help us determine the health status of the gut microbiome uh, using the deviation in the some specific gut microbes in our gut. So for that, before I go further, I would like to mention that the work I will be presenting has already been published in 2020 in Nature Communications. So to develop the index to predict the health status of the gut microbiome in humans, we collected whole genome shotgun metagenomes from published 55 studies, which includes more than 7,500 stool metagenome samples. Then we perform some quality control of the sequencing reads and perform the sample filtration based on some exclusion criteria, such as no mentioning of subjects' health status, or the study was medication or diet intervention study, or samples were collected from the uh, age of less than 10-year age of kids. Then, after the sample filtration criteria, finally we had a metadata set of 4347 samples, which includes more than 2600 samples from healthy individuals. and remaining about 1700 samples from non-healthy individuals, which includes 12 disease phenotypes such as colorectal cancer, diabetes, rheumatoid arthritis, and obesity. Then, using a prevalence-based strategy, we identified health-associated and disease-associated microbial species in the gut microbiome from using our metadata set of 4347 stool metagenome samples. And in our analysis, we considered a total of 313 microbial species to identify the health associated and disease associated microbial species. Here you can see the list, a truncated list of 313 microbial species. The second column indicate second values in the second column are for the prevalence of microbial species in healthy individuals, and in the third column values are for prevalence of microbial species in non-healthy individuals, and difference column indicate the difference in prevalence of the microbial species between healthy and non-healthy individuals. So based on differences in prevalence between healthy and non-healthy individuals, we identified these uh, identified health associated and disease associated microbial species. So in this analysis, if the if the prevalence of a species was higher in healthy individuals compared to non-healthy individuals, then we consider that species as health prevalent or health associated species. And if the prevalence of microbial species was higher in the non-healthy individuals compared to healthy individuals, then we considered the species as a health scar species or disease associated species. So based on this analysis, we found a total of 50 microbial species associated with the health phenotypes. Among them, 7 were found as health prevalent species, means which was higher in prevalence in the healthy individuals compared to non-healthy individuals, and we also found the 43 health scar species or disease associated species. Here is the list of all 50 microbial species. Then relative abundances of these 50 microbial species and prevalence of these 50 microbial species we put into a complex mathematical formula to formulate a index for predicting the health status of the gut microbiome. I am not going into the detail of detail of this formula because due to time constraint. So, but in simple words, this is the log 10 ratio of collective abundance of 7 health associated microbial species to the 43 health scar species or disease associated species. If the collective abundance of health prevalent species was higher compared to higher than collective abundance of health scar species, then value of H will be positive and the sample will be predicted as healthy. And if the collective abundance of health scar species was higher than the collective abundance of health prevalent species, then value of H will be negative and sample will be predicted as non-healthy. And this H index we call as gut microbiome health index, that is GMHI. Then, and this GMHI index ranges from -5 to +5. Positive GMHI scores represent the healthiness of the gut microbiome, and negative GMHI scores represents the non-healthiness of the gut microbiome. So we all want that our microbiome should score 5 or towards 5. Then we calculated the GMHI score for all the 4347 stool metagenome samples on which this GMHI index was developed. And here you can see that y-axis represents the gut microbiome health index GMHI score, and x-axis represents the phenotype of the sample 4347 samples, healthy and non-healthy. And the distribution of GMHI score was found to be significantly higher in healthy individuals compared to non-healthy individuals. Then we calculated the balance accuracy and found, which was found about 70%. So here balance accuracy is the balance accuracy indicate the accuracy accuracy of predicting the health status of gut microbiome using GMHI score. How accurate GMHI can predict the health status of the gut microbiome. So then we thought maybe GMHI might be working well on the training data set of 4347 microbiome samples on which this GMHI was developed. And if a new gut microbiome sample appears, it may not work well. So for that, to validate and check the sanity of the GMHI index, we considered a total of 679 gut microbiome samples from 9 independent validation cohort, which were not the part of original 4347 samples of the training data set. And then we calculated the GMHI score for all these 679 microbiome samples and found that similar to the training data set, distribution of GMHI was also found significantly higher in healthy individuals compared to non-healthy individuals. And on the right side, you can see that first three blue boxes are for the three healthy cohort from the independent validation cohorts. And you can see that all of these three cohort scores mostly positive and higher than the other remaining data sets. And all remaining non-healthy cohorts scores mostly negative. And we also calculated the balance accuracy for these 679 samples of independent validation cohort, which is higher than the balance accuracy we got for training data set of 4347 samples. It indicates that the reproducibility and robustness of the GMHI is quite high and good. So using GMHI, now we can predict whether your microbiome is healthy or not. Before this, we did not, as we know, we did not have a way to identify or predict whether my microbiome is healthy or not. We could only know that what are the microbes live in our body, but there was no particular measurement which could calculate the status, like we can calculate diabetes, we can calculate many other disease parameters based on urine test, blood test. So in some way, we are not claiming that GMHI has solved all the problems, all the issues of the microbiome field, but it's a big first step towards taking the microbiome research from laboratory to the clinics. So with that, I would like to thank all the members of the Dr. Jameson's group, funding sources, and our collaborators. And also, I would like to mention that as this is a pre-recorded talk, so if if someone has any question about my work or GMH index or anything, they can reach out to me on my email ID. I would be happy to answer all the questions if you have. Thank you.